Assalamu alaikum everyone, and welcome to the second part of gram-negative bacteria. Make sure you check out the first part of this video before watching this, because it will help you understand things better and in a more clear way. So without further ado, let's begin. First on the list today is Shigella. Shigella is a gram-negative rod, non-motile, non-lactose fermenter, and is also negative for oxidase, and does not produce hydrogen sulfide on TSI agar. TSI stands for triple sugar iron. We will know the importance of knowing this later on. So you can think of Shigella as being negative to everything, and this is what will distinguish it from other bacteria. Now in McConkey's agar, Shigella will grow in colorless colonies, since she is a non-lactose fermenter. In addition to that, Shigella grows in green colonies on Higtone's agar. As for the virulence factors, Shigella has endotoxin, since as you already know she's a gram-negative. However, Shigella does not have H antigen or the flagella, and that's why she can't move. But don't let that deceive you, because she has an awesome inbuilt mechanism to move and invade your intestinal epithelium, which we will talk about in a moment. Now, some species of Shigella produce Shiga toxin. Shiga toxin damages cells by binding to the 60S ribosomal subunit and inhibits the synthesis of protein. Now let's talk about the clinical importance. Shigella infection is called shigellosis. Makes sense. And it's characterized by acute bloody diarrhea. Let's discuss the pathogenesis of that. First of all, Shigella induces its uptake by M cell, found in between the intestinal cells. M cells are specialized epithelial cells which continuously sample particles from the gut lumen and deliver them to the underlying mucosal lymphoid tissue, where immune responses can be initiated. After that, Shigella is released from the M cell and gets into the cytoplasm of intestinal epithelial cells from its lateral side. Then it replicates and induces depolarization of actin filaments found in your cells in a way that propels Shigella to move from one cell to another like a rocket. That is really smart if you ask me. During the process, Shigella damages the cells and induces inflammatory cytokines release and recruitment of neutrophils. Therefore, Shigella infection will result in inflammatory diarrhea with blood and neutrophils in the stool. The second important clinical relevance is something related to a species of Shigella called Shigella dysenterii that can cause hemolytic uremic syndrome, or HUS, in addition to the bloody diarrhea. HUS is characterized by a triad of hemolytic anemia, acute kidney failure, or uremia, and a low platelet count. How does it do that? Well, Shigella dysenterii produces Shiga toxin, and this toxin causes damage to the endothelial cells, including those found in the glomerulus of the kidney. This will result in the acute kidney failure, or the uremic part of the syndrome. The damaged endothelial cells lead to activation and aggregation of platelets, and this will decrease the platelet count circulating in the blood. Then this aggregation of platelets will form clumps that will damage the circulating RBCs, and this is the anemic part of the syndrome. Do you remember another gram-negative rod that produces toxin similar to Shiga toxin and results in HUS too? Take a moment to think. Yes, enterohemorrhagic E. coli or EHEC. Lastly, you need to remember that HUS occurs mostly in children who are less than 10 years old. So, that's it for Shigella. Please don't forget to like, share, and subscribe, and make sure you hit the bell so you don't miss our next video in the microbiology series. Thanks for watching.